Um, First off, I really did appreciate Edward saying that Dundee's the sunniest city in Scotland. I'd like to start by saying I'm actually from Edinburgh originally, but I'm working Glasgow, the rainiest city in Scotland. Right, so I'm going to be talking about mitochondria, and my interest in these really came from, I don't know why we've got that green line there, but it should be okay, but let's go with this. You know, mitochondria, as you all know, are required for life. Primarily, the powerhouse of the cell, they produce ATP. They're involved in many, many biosynthetic reactions. They're also essential for apoptotic cell death. Now, we've known this for many years. I've been interested in this in the context of cancer. So cell death can be mitochondrial apoptosis. There's many different forms. You've probably seen every month or so, there's another paper comes out in some like Cell that will call another osis. Ferroptosis is one of the kind of hot forms of cell death just now. But cell death works to both prevent cancer and treat cancer. Tumor suppressors like p53, one way in which they act, they trigger cell death. That stops the cell becoming transformed. The vast majority of therapies, we kind of neglect this a little bit, but ultimately they work by killing cancer cells. This can be targeted therapies we heard earlier from Helen about RAS inhibitors, immunotherapies as well, and newer targeted therapies. Ultimately you want them to kill the cancer cell. A dead cancer cell, there's no way in which that can come back. So we work in different forms of cell death in the lab. The primary one we work on, which is behind me here, is mitochondrial apoptosis. Now, this is really well described. Back in the mid-1990s to late 1990s, it was really well elucidated. I think it's kind of crazy to think, ultimately, the organelle that powers our cells is required to kill the cell as well. And very schematically, what happens is, I don't know if I can get rid of, oops, no, that's not going to work. You guys, paint. Is Kurt Anderson here to help with this? I can't click away that. Okay, are you guys okay with that? I'll just kind of talk through the titles as we go through. Right, okay, so death signal. Now, cancer, that could be chemotherapy, radiotherapy, immunotherapy. You activate two key proteins in the process called BAX and BAC. Now, when they're activated, what happens is normally the mitochondria is intact. You've got the outer membrane, the inner membrane, and the matrix shown here. Activated BAX and BAC drive mitochondrial outer membrane permeabilization, really well understood process. That causes release of all soluble intermembrane space proteins non-selectively from the mitochondria, brings about caspase protease activation. Chief amongst these proteins that's involved in this is cyclin C. Again, that's really well understood. We get massive substrate cleavage through these caspases. Thousands of different proteins are ultimately cut in the cell, and then you get rapid apoptotic cell death. And so that is shown here. Just bog standard HeLa cells. They're colored in different ways, but the main thing I wanted you take home here is the mitochondria are colored here in red. It's a intermembrane space protein, M-cherry tagged. When the mitochondria permeabilize, all hell breaks loose in the cell because we get caspase activation. The cells will shrink. We'll get this membrane bleb through Rho kinase activation. And ultimately, we get flipping of phosphatidylserine with a lipid that's on the inside of the plasma membrane. As the cell dies, it's exposed on the outside. We can detect that with an X5. The cells will go purple, and then ultimately in our bodies, these cells would then be quickly removed. So just to kind of hammer home, the key point is mitochondrial outer membrane permeabilization. You see there, we get caspase activation. Eventually, an X5 labels these cells, and in our bodies, at this point, macrophages come along and eat these cells. So we understand this process to such a degree that we now have inhibitors that trigger it. They target BCL2, anti-apoptotic proteins. These have been clinically approved several years ago now for chronic lymphocytic leukemia, and more recently for acute myeloid leukemia or certain classes of it. Just to show you an example of how well these inhibitors work, these are treatment refractory patients that had CLL, chronic lymphocytic leukemia. They're treated with rituximab. In the absence of the BCL2 inhibitor, unfortunately, there's relatively rapid progression, progression in the absence of the inhibitor. You can see very nicely, however, when you treat with venetoclax, the BCL2 inhibitor, we now get this really prolonged survival. So far so good. Target mitochondrial apoptosis. We've rattled on about this in our papers for many years. It'd be a really effective cancer treatment. It turns out it is. Problem with this is, as with many targeted therapies, cells often escape this targeted therapy. However, my lab has been very interested in what I think has been overlooked in the literature for many years, is that cell death can also have oncogenic potential. Sorry, let's read it under there. Whereby cell death or processes associated with cells activating pro-apoptotic signaling can actually promote cancer and negatively impact treatment response. So we really started addressing this question: how and when can cell death promote cancer? When is that important? And really on the back of that, we've kind of stumbled into a way in which we think we can improve cell-killing cancer therapy. So first off, key event in as the cells dies is MOMP. MOMP is a binary all or nothing event. So this was thought for many years to be the case. When a cell decides, if you like, to die, all the mitochondria rapidly permeabilize. And these are MCF7 cells. Again, they express a green fluorescent protein, this time to the intermembrane space. We trigger mitochondrial apoptosis with UV treatment. You can see very nicely over the course of a 10-minute window, all the mitochondria permeabilize in this cell. So mitochondrial apoptosis, binary all or nothing event, backs and back activation, complete MOMP occurs. I'm a big Star Wars fan, and you can always take some kind of Star Wars analogy when you're thinking about most things, biology included. But I've always thought that only a Sith can deal in absolutes. Now, going from that, we then started to think, well, maybe MOMP isn't always complete. And this is work that was pioneered by Gabriel Icahn. He has his own lab now in Lyon. Kai Chow has his lab in Beijing, and Kak Hua, who unfortunately for her is still working with me. So we started to ask this question: can sublethal apoptotic stress engage MOMP in some mitochondria, so-called minority MOMP? The idea being here is we get sublethal apoptotic stress, activated backs and back. Some mitochondria would permeabilize, yet the cell could tolerate this and survive. We kind of scratched our head for several months about, could we actually image this process? And we developed a new approach, or Gabriel did, making use of a dimerization system that has been, at the time had been published already for a few years. And basically, it's a heterodimerization system. The idea being here, to specifically light up mitochondria that have been permeabilized, we'd have a cytosolic GFP with an FKBP fusion domain on it. On the mitochondrial inner membrane, we'd have M-cherry, which have the reciprocal heterodimerization domain FRB. Intact mitochondria, you've got the outer membrane, prevents these two interacting. However, when we get mitochondrial permeabilization, in the presence of dimerizer, the GFP should relocalize onto the mitochondria. And essentially what we found was, or what Gabriel found was, that regardless of the apoptotic stress, if you dialed any of these down to a low enough level, we would get minority MOMP. You can detect that, well, show that quite nicely in this cell here. BCL2 inhibitors, beach, they're only proteins, various apoptotic stresses. As long as they weren't killing the cell, we could trigger minority MOMP. So what? Okay, now to cut a long story short, what we found was that now non-lethal apoptotic stress can actually engage caspase-dependent DNA damage. And I'll show you 2 bits of key data from the paper there. Non-lethal apoptotic stress activates backs back. We get minority MOMP, the cells survive, and we could show that in different settings, yet these activate caspases. Now, one of the hallmarks of apoptotic cell death, I'm sure maybe some of you remember from your undergrad days, is that the DNA, the nuclear DNA fragments. That's dependent on activation of DNAs called CAD, caspase-activated DNAs. Now, we found that if you could trigger this under minority MOMP conditions, CAD would then cause DNA damage. Just some examples here. We can just show it again, bog standard HeLa cells. If we throw in BH3 mimetic at levels that's not killing the cell, so we're engaging minority MONP, we get DNA damage. We can completely block that with a caspase inhibitor QVD. We can directly look at DNA damage by making use of COMET assay, and again, we see evidence of DNA damage. And if we apply these BCL2 inhibitors to mice, we see evidence of DNA damage. This was work done in collaboration with Daniel Murphy at our institute, and we see DNA damage there as well. And so repeated engagement of minority MONP actually promotes transformation and tumorigenesis. Just an example here, this is a soft agar assay to look at transformation. We repeatedly treat primary MEFs with BH3 mimetic. We can then trigger transformation in these cells. We then implant the cells into an animal, and maybe not surprisingly, they more readily form tumors. So what this would suggest, and I think argues, is while mitochondrial apoptosis engaging this is a really powerful tumor suppressor mechanism, that's clearly been shown by many labs. I think we've got good evidence of this as well. Many therapies engage this to treat and eradicate cancer. If you don't fully engage the process, you've got a bit of a problem. Cells survive, they sustain caspase and CAD-dependent DNA damage, and this can be oncogenic. So this was all great. We, I think made a big splash with this. We actually got the front cover of Molecular Cell, this first time ever. I was very pleased with this. I took this to my colleague, longtime colleague, Jim Norman. He said, "What do you think of this?" He goes, "Oh, it looks like a Christmas pudding." And so it brought me back down to earth. Anyway, so we started working on this more extensively. Many labs have over the years. And it turns out that non-lethal apoptotic stress through engagement of minority MONP can have various pro-oncogenic effects. It can cause DNA damage. We've shown that, other labs have shown this. It can promote metastasis. I'm happy to get in the details of this, although it's not our own work. It's Gabriel Icahn when he started his own lab. And more recently, work that we've been involved in Matt Hangar's lab in UCSD, San Diego, have shown quite nicely therapies that engage minority MONP can actually promote survival of that cell to subsequent therapeutic engagement. So it may be a way, and I think there's still more work to be done here, to actually allow cells to, or cancer cells to resist therapy. So the tagline here is, what doesn't kill you from a cellular death point of view can actually make you stronger. It can also make the cell weirder. And I think this is a kind of reappraisal of the way we think about therapies and how they treat cancer cells. It was always like, yeah, death, no death, the cell survived. But actually the cells that can survive can undergo phenotypic changes as well. And so in the back of this, this idea that caspase activity may be oncogenic, we thought many years ago now, or several years ago now, can we actually inhibit caspase activity, or shouldn't we inhibit caspase activity when killing tumor cells? Now, still, when I present this, this seems like ridiculous. You all know, again, from your textbook knowledge, that caspases are engaged in apoptosis. They're required for cell death. In the mitochondrial pathway of apoptosis, widespread MONPs, normally all those mitochondrial perimerize, you could see that quite nicely earlier on in those movies, that really represents the point of no return. While caspases are activated and drive apoptosis, you block caspase activity, you get what we call caspase-independent cell death. And this is shown in this movie here. These cells have been treated with caspase inhibitor. We've transfected a protein called TBID, which will engage mitochondrial outer membrane permeabilization. The cells stick around in the first few hours, so this is ours up in the corner here, but ultimately every single cell in that dish will keel over and die. Very messy death. You don't get this very nice blebbing and cell shrinking. So on the back of this idea that caspases may be oncogenic if activated at low level, we thought, well, would it be better to kill cancer cells via caspase-independent cell death? Evangelos Giampazzolia, so he started this lab, or started this work. He now has his lab in Manchester, CRUK Institute. You can see he's very happy. Asma Ahmed carried on. She's not so happy, but we'll see how it goes with her paper pretty soon. But basically, they've been working on this idea, triggering a different form of cell death, blocking caspase activity. Is there any benefit? Now, I wouldn't belabor the point unless there was. What was really, really surprising, what we're finding in different tumor models, and I'm not going to go into the nitty-gritty of this approach here, But this is a prostate cancer model in which we can trigger either apoptosis in the prostate cancer. It's an orthotopic model, so the tumor is in the prostate, or we trigger caspase-independent cell death. I initially thought they would, have the same therapeutic outcome. Caspase activity may be oncogenic, but I thought it'd be rather slow. We just wouldn't see anything. But this was really surprising. We engage caspase-independent cell death in these cells, in these tumors. We often get complete regression. Now, to me, that was really shocking. Now, I'm not an immunologist, but that was suggestive that there may be an engagement of the immune system. This is only seen in immunocompetent animals. And right enough, if we now deplete CD8 T cells, so we do this by anti-CD8 depletion, we now go from the control where we see this really beneficial therapeutic effect. You deplete CD8 T cells, and then you bring this back to baseline here. So this really raised the question, why are we getting a difference in potency between the two modes of cell death? So apoptosis, one thing it is considered, and I think this holds true in most cases, it's a silent form of cell death. We get billions of cells in our bodies, neutrophils, T cells, a lot of epithelial cells in our guts as well. They undergo apoptotic cell death. It's silent. It makes sense that we don't respond to these cells. What we did find, and many other labs have shown this as well, Richard Favel's lab over in Yale, Ben Kell's lab initially at the WEHI in Melbourne as well, showed very nicely if you block caspase activity, as cells undergo caspase-independent cell death, they activate inflammation. So caspases, while dispensable for death, what they do is they block the inflammatory response of the cells as they die. And there are many ways in which that can happen. I think primarily it's because permeabilized mitochondria, if we just do RNA sequence cells that are undergoing caspase-independent cell death versus control, you look at a kind of GO-term analysis. Many of these GO-terms are associated with pathogen recognition, pathogen defense, and so forth. Permeabilized mitochondria may relate to their bacterial ancestry, elicit an antipathogen-like inflammatory response. And we know now that you need to engage these signaling pathways as the mitochondria permeabilize to trigger the immunogenicity of caspase-independent cell death. So we'll delve into activation of one of these pathways in a minute. NF-kappa B, master pro-inflammatory cytokine, CGAS sting. So somehow we still, I would argue, don't know how this works. Mitochondrial DNA is released from the mitochondria at one point, and I still think that's unclear. And MDA5, you can get double-stranded RNA produced as a result of the bidirectional transcription of the circular genome within the mitochondria. So to summarize this part, caspase-independent cell death beats apoptosis at inhibiting tumor growth. So I think we're now appreciating it's not simply whether a cancer cell or any cell dies for that matter that's important, but beyond that, how that cancer cell dies can have really consequential effects upon the immune response. Therapeutic effects of engaging caspase-independent cell death requires intact immunity. And so killing cells under caspase inhibition may be an effective anti-cancer therapy. And I like this has been a really fun project because we've gone from very fundamental discovery science. We're now collaborating, or Cancer Research Horizons is doing quite a lot of work now to explore this as a novel anti-cancer therapy. So that may be one way in which we can engage MOMP-induced inflammation. The other aspect of the lab is being really interested in understanding, does MOMP also have non-lethal inflammatory function? So kind of pairing this idea that we get minority MOMP, that can, you get cell survival, can it also have inflammatory effects? We've shown, or a couple of years ago now, that it can promote the inflammatory phenotype of senescent cells. That's worked together with João Passos. Other labs have shown various roles for innate immunity, dendritic cell activation, chemosensitivity as well. And so this, I think, suggests a minority MOMP, besides causing DNA damage, can also have inflammatory effects and trigger various processes. So mechanistically, how does MOMP signal inflammation and immunogenicity? And this is really the last part of my talk, and this work led in the lab by Joe Riley, who's got his lab now in Innsbruck. Esme Vringer moved back to Amsterdam recently, and Rosalie Heilig, who's moved back to Switzerland recently. And the work comes from a paper we published a couple of years ago in Embo Journal, which showed that as mitochondria permeabilize, and so here we've thrown on BH3 mimetics, BCL2 inhibitors, along with caspase inhibitor, and then pulled out the mitochondria, we can see extensive mitochondrial ubiquitination. You can see that either by Western blot, we can also see that through imaging. And initially we thought, well, okay, what's that ubiquitination doing? Maybe it's serving as a signal to target those mitochondria for mitophagy. This has been seen extensively in the case of pink-1 Parkin-mediated mitophagy. You get ubiquitination mitochondria and that removes them or serves as a signal to remove them. However, we never saw any role for autophagy in removing these mitochondria. So we kind of start to look elsewhere. We delved a little bit further into the ubiquitination. We can do mass spec-based approach to understand what proteins are being ubiquitinated, shown here in pink. The vast majority of these fall in MOMP localized to mitochondria, maybe not surprisingly. If you look then at the ones that are upregulated in ubiquitination, they span both the outer, the inner, and even the matrix, suggesting there's some intermembrane rupture. And there are many, many different proteins that are being ubiquitinated. There's over 200. So whatever is doing this, it's very promiscuous. So ubiquitin chains have very distinct biological functions. The kind of classical one, K48, serves to tag and send proteins for proteasomal destruction. We saw some evidence of that, but interestingly, through data I'm not going to show you just now, but we also saw K63 and M1 linked ubiquitin. Now, these were very interesting in the context that they act as signaling linkages. So they connect. Once this happens, you basically can stimulate inflammatory responses that I'll delve into in a minute. So we saw that on the mitochondria once they're permeabilized. This was interesting because work done by others, Felix Randau at the NLMB and Ivan Dikic several years ago now, showed very nicely that as salmonella invade cells, they get ubiquitinated. That serves as a signal to attract this adapter molecule called NEMO, which is involved in NF-kappa B activation. And by doing so, you then, or the bacteria then stimulates a cell-autonomous innate immune response. You get activation of NF-kappa B, protecting, or at least partly protecting the cell from infection. Now, again, given the bacterial ancestry of mitochondria, we then ask this question, does MOMP activate an analogous pro-inflammatory response? And indeed, that's what we found, either by imaging or by Western blot of the endogenous protein. We found that NEMO was recruited to mitochondria after MOMP. Gave rise, the kind of take-home message of this paper was, once MOMP occurs, we get extensive mitochondrial ubiquitination, NEMO recruitment, and activation of pro-inflammatory NF-kappa B signaling. So many questions arise from this, one being what E3 ligases are required for mitochondrial ubiquitination. We looked at various kind of, targeted hypothesis, pink-1 Parkin, CAP1-2, proteins, E3 ligases that are known to be at the mitochondria. None of this worked. And, I just said to Rosalie, you've only got another 600 E3 ligases to go through. And she told me to do one. So we thought about doing another approach rather than knocking them out individually. So I'll just finish off with thanking the lab. This is the current lab. Shown here, I think I've highlighted most of the people who've done the individual bits of work. It's really a phenomenal honor to receive this Hooke Award. I look back at the previous recipients. It's really a stellar award. I'm really pleased about that. I think, I told my kids I was getting this, and they said, "Well, you never win anything." They give you straight up, as you might imagine, but they're pleased for it as well. But it's really, I'm just kind of tip of the iceberg. It's thanks both to my lab, but I think equally important, colleagues, collaborators, mentors, and funders, of which there are many. And if you start listing them, you invariably forget them. So with that, that's my lab. I should also thank my family. They give me a lot of grief over the years. Equally, I give them grief, vice versa. And a big shout out to my wife. I get to go to a lot of conferences. I'm away a reasonable amount of time, and she's holding the fort back home. So I very much appreciate that. I think last off, I got sent a last-minute slide to include about Company of Biologists, just to say it's a great organization, great charity. I'm a trustee director of it now as well. I think all All I would say is that the money that goes in there is then redistributed in the form of workshops and grants. It goes back to the community as well. The basis of the funding for it is through the journals. So I can just encourage you, obviously Journal of Cell Science is a relevant one here, but encourage you to consider these journals when thinking about your next piece of work. Okay, so with that, maybe got a couple of minutes. Happy to take some questions. Thank you.